pediatric lungs.
What number are you?
I am number 1110. Yes, I'm number 1110. Before I start my presentation, I want to tell you a bit about the significance of this project. For many people suffering from end-stage lung disease, the only treatment option available when all other treatments have failed is a lung transplantation. Unfortunately, there is a shortage of lungs available for transplant. Many of the lungs that do become available are too damaged that they don't meet the organ procurement requirements and are simply discarded. Rather than discarding these lungs, we propose that they can be used to process acellular human lung scaffolds. We have previously developed in our laboratory a process that removes all the cells from the tissue, all the blood, but retains the tissue-specific extracellular matrix. This matrix, specific to the lung, has the elasticity and strength of the gross architecture to support lung tissue formation. These scaffolds, these whole lung scaffolds, also possess pre-existing vascular and bronchial tree networks that can provide the framework to rebuild some lungs. The purpose of this study was to determine the feasibility of using these lung scaffolds to bioengineer human pediatric lungs. These images show the decellularization process. So the image at the top is a pediatric human lung, and the white one in the bottom is an acellular human lung scaffold that has no cells. So what does a normal lung scaffold look like? So this is a multiphoton microscopy image of a normal lung extracellular matrix. Multiphoton microscopy was used with autofluorescence was used to image the collagen bundles, shown in green, and the elastin fibers, shown in red. The image down here at the bottom is a cross-sectional view of the same image. So the scaffold is made up of the extracellular matrix of the tissue, and the extracellular matrix is mainly composed of the collagen and elastin. Before we could attempt to bioengineer lungs, we had to come up with a cell installation protocol. So to do this, we first decellularized single human lungs, as seen in this picture right here, and we used cell lines and immortalized human cells, human lung cells, to examine cell numbers that we needed, culture conditions. We used these bioreactors to remove cells and also to add cells back into these lungs, and so we needed to figure out what the perfusion rates would be needed to do this. Once we established a protocol using these single lungs, then we moved on to our pediatric double lung sets. We had 3 of each, so 3 single lungs and 3 pediatric double lung sets. For these installations, we used primary human lung cells isolated from viable human adult lungs. And so the cell mixtures that we had for these primary cells were alveolar epithelial cells, vascular cells, specifically lung vascular cells, and tracheobronchial cells. We wanted to examine what the cell number and the pre-treatment of scaffolds would do to tissue formation. So to do this, we modeled this in a smaller scale of our scaffolds, and so we have here the picture of the lung scaffold. So we cut this into pieces, as you see in these pictures, and we modeled this in these pieces. So we divided some of our pieces into groups. One group was untreated, the other group was treated with human serum, and the third group was treated with platelet-rich plasma. Each of these groups also received different numbers of cells. Some groups received 100,000 cells, others received 500,000 cells, and the final group received 1 million cells. What we found is that treatment with serum and PRP resulted in better tissue formation, but treatment with platelet-rich plasma always had better tissue formation compared to serum, regardless of the number of cells used. And so we decided to use platelet-rich plasma because it provides a lot of growth factors for the bioengineering of our tissues. This table summarizes the cell installation protocols we used. So as I mentioned before, we started with single lungs 1 and 2 and 3. Here we describe the cell types used, the viability of the cells, the number of cells installed, the method of installation. So depending on the cell type, they were either installed with the trachea or the artery. Also the flow rates of installation and flow rate between installations. The main difference here is that once we established our installation protocols using the single lungs, we then altered our pediatric lung cell installation based on data we received from the single lungs. And so the only difference we changed for our pediatric lung installations was that we increased the number of cells, and we also slowed down the flow rate to allow the cells more time to attach. So this histology shows the histology for the bioengineered pediatric lungs. The top row, A through C, is histology for a normal pediatric lung, to use as a comparison. So this black arrow is pointing to a blood vessel that contains red blood cells. Images D through F show pediatric bioengineered lung 1. Image F shows a bronchial airway, and it shows the presence of ciliated epithelial cells. Image I shows a blood vessel as pointing to a blood vessel, but as you can see, it doesn't have any red blood cells because this was the lung we bioengineered. There was no blood in this lung. It does have a hydrogel mixture that I will talk about in the next image. Images K through L are pediatric bioengineered lung 3, and image L shows repopulation of a blood vessel within one of the lungs. So I talked about this hydrogel. To evaluate the vascular patency in these bioengineered lungs, we injected this hydrogel chloronic with a rhodamine dye, a rhodamine red dye, into the artery of the pulmonary lungs, of the bioengineered lungs. What we found was that this hydrogel was contained, as you can see in this image, all those red areas where the hydrogel was, it did not leak into other areas of the lung, it was contained within the capillaries. We also stained for CD31, which is an endothelial marker. And so over here in this image, you can see that wherever we had this hydrogel dye, endothelial cells surrounded the capillaries. So all these capillaries were bound by endothelial cells, which is what you would see in a normal lung. This is an electron microscopy image of this bioengineered lung. You can see that we have the presence of type 1 alveolar epithelial cells. We also have type 2 alveolar epithelial cells. And here we see some capillaries, and as I mentioned before, these are empty. They have no red blood cells. There was no blood in this lung. We examined the expression of lung-specific proteins in our lungs. So we examined pediatric lungs 1, 2, and 3. We compared the expression of these proteins using normal lungs. So these are 2 separate normal lungs we used. We examined the expression of aquaporin 5, which is an alveolar type 1 epithelial cell marker, surfactant protein C, and surfactant protein D, which are important for lung function. As you can, beta-actin was used as a loading control. As you can see, expression of aquaporin 5, SPC, and SPD was very similar to their expression in normal lung. To examine the collagen content in the lung, here we use multiphoton microscopy and second harmonic generation imaging again. So these views right here, the normal shading view and the surface mapped view, show the structure and the collagen content of the acellular scaffold. And once we added cells back into our scaffolds, you can see that there was an increase of collagen in our recellularized lung scaffolds. Quantifying the amount of collagen using second harmonic generation imaging, we were able to see this and prove it. Our recellularized lungs had significantly more collagen than the acellular lung scaffolds. Conclusions. So what we found was that our bioengineered lungs had both type 1 and type 2 alveolar epithelial cells, like a normal lung would. We had the formation of alveolar capillary junctions. We also had the production of surfactant protein C. And our bioengineered lungs had more collagen than the acellular scaffolds. So this is great. But we still have a question. Does this process work to really make a functional lung? What we did next, so using the same installation protocols we did for the bioengineered lungs, we produced a pig lung. And so we had 2 lungs to do this. One pig lung was used to create a scaffold, which you see here. Another lung was taken from a pig, so this was a single lung pneumonectomy. We isolated cells from this pig, as you see here. We combined the scaffolds using these primary cells, and we bioengineered a pig lung, and this was cultured for 30 days. What you see here is pig 1, Harry. This is the first pig that received the first bioengineered pediatric lung we had. So this is 10 days after his transplant. He had no complications, and he seemed very normal. He was fine. This is the electron microscopy image of the bioengineered lung post-transplant. So the one I showed you before is pre-transplant. This lung, you can clearly see that there are more cells. Here we also see the presence of type 1 cells. We still have type 2 cells, but now our capillaries, you can see, are full of red blood cells. The red blood cells are stacked within the capillaries. Here we also focus on an image showing a type 2 alveolar epithelial cell. So that's the image comparing a normal type 2 alveolar epithelial cell with our bioengineered lung showing a type 2 cell. And as you can see, they look very similar. So this study shows the first attempt to produce a bioengineered lung. This will provide the groundwork to hopefully achieve our long-term goal of bioengineering lungs that can be used for transplant. I would like to thank the Nichols Cordiella Lab, my mentor, Dr. Joe Nichols, and my co-mentor, Dr. Cordiella, our lab manager, Jean, and one of my colleagues, Yesenia. Michael Riddle is the person who designed the bioreactor we use in our lab. Gracie Vargas and Raul Paul are the ones in charge of helping us with the multi-photon microscopy imaging. The people from Methodist Research Institute Hospital, Dr. Saverio Francesca is the cardiothoracic surgeon that does the transplants for us in our pigs. We are currently doing our 4th pig transplant. Jessica Rudy helps with the decellularization procedures for the lungs, and I would also like to thank my dissertation committee members. And with that, I will take any questions.
So these scaffolds are single use only, yes?
Single use is in just, we use them to seed and then can't use anymore?
Yeah.
So we usually, yes, use them only one time. What you can do is isolate cells back from that scaffold, but once you use it to isolate cells, then yes, it's